Good afternoon, ladies and gentlemen. Welcome to my presentation. I appreciate the opportunity to be here with you today. I'm Hsin-Wei Kuo, a researcher from National Pingtung University of Science and Technology in Taiwan. The topic of my presentation is Valorizing Agricultural Waste, Cocoa Pod Husk for Sustainable Aquaculture, Achieving the Sustainable Development Goals. Today, I will first discuss about cocoa industry. I will then touch on aquaculture of white shrimp, and finally, describe how application of cocoa pod husk on immune response of shrimp. Cocoa is commercially grown in South America, and its beans are primarily used for chocolate and cocoa-based food production. The consumption of this product has been steadily increased worldwide. The global cocoa production was estimated to be 4.7 million tons in 2019 and 2020. In cocoa industry, only beans were used to produce the product of chocolate. Apart from the cocoa beans, cocoa pod husk was considered as an agricultural waste in the past. Nevertheless, the cocoa pod husk, which account for 67 to 76% of cocoa weight, are mainly byproducts of the cocoa. However, the cocoa pod husk, which account for a significant portion of the cocoa pod weight, are mainly considered a byproduct. In the past, cocoa pod husk was treated as an agricultural waste, resulting in the generation of a large amount of cocoa pod husk every year. Improper management of cocoa pod husk leads to environmental pollution and the spread of the disease, such as the black pod, which is a major cocoa disease. It is causing significant losses in the industry. However, studies have indicated the cocoa pod husk present potential and renewable resource in various industries, such as food, fuel, dermatology, and medicine. Developing processing methods to unlock the diverse function and bioactivity of cocoa pod husk is a promising strategy to enhance the value chain in cocoa production. Bioactive compounds extracted from cocoa pod husk can exert potential health benefit and may therefore be an alternative source of this important component. These bioactive compounds including the pectin, antioxidant, dietary fiber, and minerals. In present study, I will describe how to use the cocoa pod husk in the aquaculture industry. The aquaculture industry has experienced rapid growth and plays a crucial role in providing high-quality animal protein. With population increase and change in eating habits, humans' demand for protein is higher than before, especially in Asia. The demand for protein is increasing about 349%. Aquatic products are highly quality source of animal protein. Aquaculture grew faster than any other food-producing sector. It will supply more than 50% of aquatic food consumption in the future. In the past, antimicrobials and chemotherapies were extensively used to maintain the health of aquatic animals in intensive culture. However, increasing evidence suggests that the use of antimicrobials in aquaculture can hinder immune response, leading to the accumulation of residues in aquatic animals, contaminate the culture environment, and contribute to the emergence of drug-resistant bacterial strains. To address this issue, natural immunostimulants derived from polysaccharides extracted from herbs, plants, or seaweed have been utilized to enhance immunity and promote growth in aquatic animals, thereby reducing the overuse of drugs. Various polysaccharide compositions, such as fucose, galactose, glucose, mannose, rhamnose, and xylose, have shown promising application in the aquaculture industry. In our research, we extract the bioactive compound from cocoa pod husk. Here is the extract method. Briefly, the fresh dry cocoa pod husk was dried for 7 days under 50 degrees Celsius, then grind into powder. Add DD water, then heat it to 95 degrees Celsius for 15 minutes. Then upturn the supernatant. After the night half-life, we store the extract at negative 20 degrees Celsius until use. Cocoa pod husk extract contained significant amounts of many types of polysaccharides. These polysaccharides make the cocoa pod husk a potentially valuable immunostimulant for the aquaculture industry. White shrimp is a highly valuable and widely cultured species in the aquaculture industry. Its strong disease resistance, rapid growth, adaptability to different environments, and short breeding period contribute to its popularity. Global production of white shrimp in 2018 accounts for over half of the crustacean population. However, intensive shrimp farming has led to the emergence of the viral and bacterial diseases, resulting in significant economic losses. To ensure the sustainability of shrimp farming, integrated approach that focus on maintaining water quality, temperature, and culture intensity, as well as enhancing the shrimp's immune response, are the major concerns. This study aims to extract bioactive substance from agricultural waste, cocoa pod husk, and inject them into white shrimp to evaluate their immune response and disease resistance. The study designs as following. First, immunity of shrimp after inject by cocoa pod husk. Then survival rate of shrimp after inject by cocoa pod husk extract under the hypothermal stress and infection of the Vibrio vaginolyticus. PO activity is the immune parameter. Propel system is non-self-recognition system and one of the most important immune mechanisms in crustacean to prevent invade of pathogens. The shrimp injected with the cocoa pod husk extract has the significant increase in PO activity after 1 and 3 day injection. The propel system related genes has a significant increase as well. It is indicated that cocoa pod husk has the promotion effect on the PO activity and the propel system. Here is another immune parameter, phagocytosis. Phagocytosis is a major and the most common reaction in cellular defense mechanism to remove invading pathogens or foreign particles in innate immune system. The phagocytotic activity and clearance efficiencies of white shroom injected with the cocoa pod husk extract has the significant increase than control group for the 1, 3, and 7 days. It is indicated that cocoa pod husk extract has the promotion effect on phagocytotic activity and clearance efficiency. The survival rate of shroom injected with the cocoa pod husk extract at 20 micrograms per shroom has the 26.6% increase under the hypothermal stress. The survival rate of white shroom injected with the cocoa pod husk extract after 1 day at 20 micrograms per shroom has 20% increase, challenging with the pathogen infection. This result indicates that injecting cocoa pod husk extract can enhance the stress resistance and disease resistance of the white shroom. In conclusion, in a present study, we explore the utilization of cocoa pod husk extract, previously considered as an agricultural waste, as a result for extracting biological activity compounds. We then apply this extract in aquaculture. The findings reveals that the injection of cocoa pod husk extract enhance the immunity and survival rate of white shroom when exposed to pathogen infection and hypothermal stress, strengthening the resistance of white shroom to pathogen and stress could potentially increase production in the shroom industry and provide a valuable protein source for human consumption. This study demonstrates that cocoa pod husk be utilized as a recycled and sustainable natural resource, aligning with the SDGs goals by the United Nations. Reusing waste materials and contributing to sustainable practice, we can work toward achieving more environmental friendly and resource efficient future. In conclusion, white shroom after injected with dry cocoa pod husk extract enhanced propel system related genes including the LGBP, PE, and propel-1 expression, upregulating immune response and increased susceptibility of shroom to Vibrio organolyticus. Also elevate tolerance to hypothermal stress at 14 degrees Celsius. All the results indicate that cocoa pod husk extract is a potential immunostimulant to apply on shroom and is a reusable natural resource. And it is the end of my presentation. Thanks for your attention.